everyone, and welcome to another session of the Rosalind Franklin Society annual meeting. I'm Juliana Lemure, science writer at Genetic Engineering Biotechnology News, or GEN, and I'll be your host for this session called Plants to People and Back. This session is a little different than the others at the meeting because the topic is plants, not people. That said, the work we'll be discussing today connects to people and to a bigger picture in the life sciences. So to get into all of this, we're joined by two very special guests. Dr. Joe Handelsman is the director of the Wisconsin Institute for Discovery at the University of Wisconsin-Madison and a Howard Hughes Medical Institute professor. In addition, she served as a science advisor to President Barack Obama as the Associate Director for Science at the White House Office of Science and Technology Policy. On top of all that, she was the founding president of the Rosalind Franklin Society. Joe is responsible for groundbreaking studies in microbial communication and metagenomics, in addition to being widely recognized for her contributions to science education and diversity in science. She's also an author and just released a new book a few months ago entitled A World Without Soil: The Past, Present, and Precarious Future of the Earth Beneath Our Feet. Joe, thank you so much for joining us today.
Thanks for having me. It's great to be here, Juliana.
We're also pleased to welcome Dr. Laura Boykin, Senior Scientific Consultant at BioTeam. So if you're one of the almost 2 million people who have seen her TED Talk, you know well who Laura Boykin is. If not, I'll fill you in. Laura is a senior TED Fellow whose research focuses on true partnerships with African scientists while using genomics to help farmers in sub-Saharan Africa fight crop diseases such as whiteflies and viruses. Along with scientists in East Africa, she founded the Cassava Virus Action Project to roll out portable DNA sequencing and analysis to farmers. The team also trains local communities in the use of these technologies to tackle future outbreaks. She's the winner of the Gifted Citizen Prize and has been named one of Wired Magazine's People Racing to Save Us from Ourselves. Laura, it's great to have you with us today.
Thank you. Can I take you everywhere? I feel so good about myself now. That's amazing. Thank you for that. Wow. Thank you.
You're very welcome. So we're going to get things underway, Joe, with a short talk from you, and then we'll hear from Laura, and then we'll have some time to have a conversation about science, diversity, being a woman in science, and so much more. So please, Joe, now the floor is yours.
Thanks, Juliana. So I was going to just talk a little bit about the fusion of plant sciences, microbial sciences, and human sciences, if we can call it that, because I think most distinctions in biology are pretty arbitrary these days. Most of what we do, at least in the life sciences, I think spreads from one to the other very fluidly. My career, I think, illustrates that. When I started, my undergraduate work was in agronomy, which is a combination of crops and soil. And then my graduate work was in molecular biology, where I studied plant microbe interactions, so the bacteria that live on plant roots and provide nutrients for them there. And then when I started my own research program as a faculty member, I pursued other kinds of bacteria that live in association with plants. And one that we discovered early on produced a very exotic new antibiotic. And it turns out that that antibiotic has created almost an entire career of studies because it's so interesting from a chemical standpoint, a genetic standpoint, biochemical, and then also in terms of its many, many impacts on biological systems, from killing plant pathogens to killing human pathogens to even augmenting the activity of BT, which is a bacterial agent that's used to control insects in agriculture. So I have kind of gone back and forth from the agricultural side of microbial sciences and plant sciences into the human. I became very interested from a personal standpoint, but also, of course, it coincided with my work with the antibiotic resistance problem that we're facing in the world today. It's a global problem. It's predicted that antibiotic resistant bacteria will be the leading cause of death in 2050. And I experienced that very personally and up close when my mother acquired bacterial infections of her lungs. It was a series of infections over quite a while, 17 years. And eventually they developed antibiotic resistance and couldn't be treated. And that was ultimately what killed her. And so there was this sort of career-long antagonism between my personal life, where for years I was watching my mother get sicker and sicker because of antibiotic resistant bacteria, and my lab, which studied soil bacteria that produce antibiotics. And I thought there has to be a way to discover antibiotics more quickly and get them to the market so that they could help people who have antibiotic resistant bacterial infections. And so most of our antibiotics come from the soil, but in the 1980s, most of the antibiotic discovery companies pulled out of the field because antibiotics aren't nearly as lucrative as things that people take like Lipitor or something like that every day for the rest of their lives. Antibiotics are almost too successful. We take them for 5 days or maybe 10 days, and then we're done. Hopefully we're cured. And so it's a real catch-22 that we're facing this global antibiotic resistance crisis, and yet we can't get the companies to move back into antibiotic discovery. So I was at UW-Madison, where I am now, until 2010, when I moved to Yale University. And I was dealing with this quest of how do we discover antibiotics in a new and more efficient way from the soil. And so I started a new course that's now called Tiny Earth. And it started with 6 students at Yale, and now is about 10,000 students a year taking the course all over the world. And the idea is that students dig up soil samples of their choice, they learn something about the soil, and then they culture bacteria from that soil and then test the bacteria for the ability to produce antibiotics. And so every student who's taken the course pretty much discovers an antibiotic producing bacterium. Of course, they're not most of them are not, of course, novel antibiotics. And so my lab does a lot of the chemistry following the initial discovery, but the students do characterization of their organisms. And the idea was to essentially crowdsource the problem and see if we could use the diversity of soils and the diversity of microbes in those soils, and then the diversity of chemicals that those microbes produce to address the antibiotic crisis. And so when I came back to Wisconsin after Yale, I expanded this program, and we now train instructors from all over the world, where I think we're in about 30 countries now. We have about 700 trained instructors who teach the course, and they teach, as I said, thousands of students each year. So we haven't discovered the next big drug yet, but we're close to having a structure of a new antibiotic. And we're really excited about this as both a novel way of doing science, incorporating undergraduate students as well as many different institutions and countries into the discovery process, and then having this very collaborative group across the world. And that in itself is very inspiring to the students. The students just love it because they're doing original research, but they're also part of something much bigger than themselves, both a whole coalition of people dedicated to a global health problem, but then also people who are doing work just like them at other colleges and universities around the world. So that's been a very satisfying part of my career, and again, straddles that soil microbiology and human disease continuum that I've sort of always been a part of. One of the things that's getting very exciting to me is that as we begin to look at these organisms from a more, in my opinion, biologically relevant standpoint, the fact that these are bacteria that live in soil and around plants, has to be taken into account in our study of them and our coaxing them to do what we want them to do. And so one of the problems that has existed forever in the antibiotic field is that we know bacteria have the capacity genetically and genomically, in some cases, to produce as many as 20 or 25 different compounds, and yet we only see one produced. And so my lab is now looking at the possibility with some really interesting data that if we consider where these bacteria came from, and the fact that they probably have pretty strong relationships with other soil bacteria as well as plant roots, maybe we can coax them to produce some of these antibiotics that are currently silent or invisible to us. So again, I talk to people in the antibiotic industry, and they screen most of their organisms, for example, on brain-heart infusion, which comes from cows. Well, the bacteria that we isolate from soil really don't see brains and hearts very often in their lifetimes. And one of the first things that one of my students at Yale discovered was that if you culture the bacteria on a medium called PDA, which is made from potatoes, potato dextrose agar, you get a much higher frequency of antibiotic-producing bacteria. And it's really funny because when I talk at medical meetings or at antibiotic discovery meetings, and I say, yeah, we cultured on PDA, they all look at me like, well, what's PDA? Well, that's something that plant people use. It's not something that is typically used in medical research or in the antibiotic industry, but they're all still discovering organisms from the soil. So I think there's been a whole realm of science that's been missed because of this split that we've had, which I think is arbitrary, between the human health side and the environmental and agricultural sciences. So we're hoping to unite those. In between my going to Yale and coming back to the University of Wisconsin, I worked with President Obama as a science advisor for about the last 3 years of his administration. It would have been longer, but the Senate took a while to confirm me. That's a whole nother story. And that was a really exciting time because I was head of the science division in the Office of Science and Technology Policy. And the science division had this incredibly broad mandate that spanned from the medical sciences to the agricultural to some of the environmental sciences, and very, very basic science as well in fusion technology, energy technology. We were responsible for making sure that the agreement was signed with CERN, the facility, the super collider in Switzerland, between the US and the Swiss government. So the breadth was amazing. And so I got to learn so much science. It was probably the most stimulating time of my life because I never felt like I was on top of the science. You know, when you switch from telescopes to infectious disease in the same day, I was dealing at one point literally with the demise of a very precious telescope in West Virginia, which we were able to save. And then the Ebola crisis, the epidemic in Africa, all in the same day. And that was the kind of juggling that we had to do. And it was just an incredible experience to deal with that kind of breadth of science and then have experts all around me who knew the fields that I didn't know as well, and could tutor me, and then I could tutor the president on those issues. So it was a really thrilling time, and it was a particular privilege to work with a president who absolutely loves science, and he loved having scientists around. He loved talking science, and there's a real special characteristic to your job when you're talking to the president of the United States and he tells you that this is the highlight of his day. You know, he's had 12 meetings before you that day, and he says, oh, science is the best part. And it was almost like that was the fun part for him. Of course, some of it wasn't so fun when it was global epidemics and things like that, but he really just loved it. So that was just an incredible opportunity that, you know, we'll always treasure, working with him and working with an amazing White House staff to learn science and to make policy around science and also for science, to make sure that science was done as readily and safely and as well as it could be done. So that was an exciting few years. And then I came back here to the University of Wisconsin to run the Wisconsin Institute for Discovery. And we're kind of remaking that in some new ways, trying to use interdisciplinary, very fluid interactions among scientists of different backgrounds and different fields to generate new discovery. And we also have diversity, both gender and racial and sexual orientation, all kinds of diversity as a very core part of what we do, because the research shows that people who are diverse, groups that have diverse or heterogeneous membership, are much more creative and come up with more solutions to problems. That's been shown for 50 years of research. And so we're adhering to that research by saying, well, okay, if we want to make new discoveries and do new science, then we need to recognize what it is that generates exciting science, and that is diversity. So we've started programs that really help us build diversity, hire in diverse ways, and also foster communities, people communities that generate diversity and support diverse people. So it's been an incredible career, very, very exciting, and in a lot of different areas, both of science, but also of where science is done, how it's done, and the institutions that are affected by science.
Fascinating. Thank you so much for sharing all of that. I could ask a million questions, but before I do, I'll pass it over to Laura, and she can tell us about what she's been up to recently, and then we'll have a conversation. So Laura, the floor is yours.
I had a title like a scientific one, and then I thought I would just, be creative here and call this Science, Social Justice, and Saving Cassava. I have a new last name now. I got married this year, so I'm excited about that. So I put that on my slide just to confuse the algorithm of tracking me scientifically. I added that to my slides as well. Okay, so I think that the transition from what Joe was saying about what matters the most in science and what matters the most just on the planet in general, for me, I care about two things really. One, these farmers that don't have food. I cannot understand why in 2022, these women don't have enough food to feed their families. I just simply cannot understand why and how we are letting that happen. So I care about them. And secondly, I care about making science less racist. I think that it's baked into science and we need to unbake it. And so the things that motivate me are the women farmers in East Africa. 80% of the farmers there are women, and empowering women to help women are the things that I care the most about. So those are the things that get me out of bed. Those are the things that are my core values. Those are the things that drive me to do science. And so with those two things in mind, I was really fortunate to be approached by a really awesome team of scientists who you can see here, all plant pathologists or plant virologists. Monica Kehoe's in Australia. Joseph Nguru, who you see taking our selfie, is an amazing plant pathologist in Tanzania. Next to him is Dr. Titus Alichai, who is in Uganda. Dr. Peter Serwaji is a Ugandan who was working in Tanzania. That's me laughing as usual. And then next to me is Professor Elijah Ateka. And this is a core group of scientists who have been paving the way. And I cannot even talk about what I'm talking about without extending my warmest thanks to them and support. So there's a big, massive group of people around the work that I'm going to share with you all. And so everything that we've worked on has been to do with cassava, which is the plant that you see here with my graduate student, Tony. And not very many people know, but cassava is actually what we call a poverty fighter. It feeds 800 million people. 800 million people a day rely on this plant for their daily calories. And that is, I mean, mainly in sub-Saharan Africa, but also very, very prominent in South America and Southeast Asia. So this plant is key. It's key to food security. It's key to the sheer livelihood of many, many, many people on the planet. And we've spent a lot of time studying it. And the problem is seen here in this picture. We have been working with scientists to increase the yield, okay, of this plant. And you can see the part that's eaten and the part that people rely on for the calories are the roots, the tubers. And the gentleman in the middle is holding a local variety. You can see that the tuber is pretty reduced there. And the two families on either side of him are holding cassava from improved variety. So these are locally bred by the cassava breeders in the region. And this was the day I went out to a field and actually saw science making a difference in people's lives. We do a lot of talking. Science, we love to talk. We think publications matter. We think all those things matter. For me, the only thing that mattered to me was this day. It motivated me to go, oh my gosh, science made a difference. It didn't matter if this was published in Science or Nature or in another journal. It mattered that it made it to the field, right? It mattered that it transformed people's lives. So this was a trial that was set up. There was one row of the local variety and then about 100 rows of this improved variety that had come from the cassava breeders. And this improved variety was resistant to the diseases that were circulating in the region. So this was the motivation for a lot of the work I'm going to talk about. And what's really causing the problems for these farmers are these two viral diseases, cassava brown streak disease and cassava mosaic disease. And the cassava brown streak disease are RNA viruses. Everybody knows about RNA viruses now. Everybody knows about this. We're in a global pandemic, but these farmers have been dealing with the global pandemic for a long time. A long time they've been struggling with these diseases that are showing up as rotten roots or constricted roots. The cassava mosaic disease is a DNA virus. And the problem is you have these white flies that fly around and feed on plants and transmit these viruses everywhere, all over the place. So we call the white flies like little dirty hypodermic needles because they pierce the underside of the plant, suck up the virus, then they go to the next plant and infect it. So you have these two viral diseases circulating around, and a farmer can go from having 40 tons per hectare to zero in a matter of a season, which is devastating for the family. So what typically happens is you have farmers, and the best part and the most beautiful part about this is the women work together on their farms. They work to plant. So this day we came out and this particular group of farmers, this is actually my collaborator's sister's farming group. And they were showing us how they were planting the cassava, and they'll plant these cuttings and then they will go to the next woman's farm and plant cuttings and go to the next woman's farm. So they're already scaled automatically to improve each other's lives, which is amazing. So they'll plant these cuttings and it takes 9 months for the plant to mature. And you can imagine that after all of that, 9 months later, you pull up the plant and this is what they get. I mean, it's almost impossible to feed your family on this. So the scientists that I mentioned in the beginning have been working a long time to try to characterize these viruses, right? One of the first things that we had to do when this global pandemic started was, what is it? What is it? How do we detect it? What do we do? And, these are the devices that were made for white people by white people. Let's be honest. This device is $1 million. It requires constant power. It requires expertise. And it really hasn't made it to a functional state on the African continent. So great, you have a disease and you have a tool that is unusable and not there. So we spent a lot of time. I lived in Australia. I had a lab. I did the whole academic thing. And then I started to realize if I'm going to transform people's lives, it's not going to be in that space. So I left that position and we, just really, and it's interesting that Joe mentioned the Ebola crisis, because it was around this time we were like, they're using other devices for the Ebola outbreak. Why are we not using these for farmers? Like, let's see what the medical field is doing. Why are we not using it? And so this is the device that replaced this big machinery, right? Technology has moved to such amazing feats that these $1 million things that require a whole room now can run off of a USB stick plugged into a laptop and fit in your hand, right? So this particular thing is called a Minion. This company in the UK makes these things, Oxford Nanopore, and it's really just absolutely revolutionized the way we collect genomic data. You can do this for anything. I'm sure people are using it on all the cool soil samples Joe talked about. They're doing it for Ebola sequencing. They're doing it for SARS-CoV-2 sequencing. And we thought, why are these farmers, the farmers we care the most about, why are we not using these to diagnose their problems, right? Because really early detection is key. So we founded a group of people. This is our group. Everything we need to do DNA sequencing is happening by what people are carrying here. We have our sequencer, we have our DNA extraction, and we have our computer to do the analysis. Okay, so I'm just going to click through and show you and I just also want to say that everybody said we could not do this. So that was cool, because I love when people say I can't do stuff, because then I just do it. And it's exactly what Joe said. It was our diverse, inclusive way that we designed the project that made us just do it. So we took everything out. We called it a tree lab because we sat under the tree, and the farmers, we walked around with the farmers. Farmers want to know, is my plant sick? Is it the rain? Is it something in the soil? Is it the virus? So this is Mama Mina. She is one of the amazing farmers that allowed us to bring all of our gadgets onto the farm. It's Brenda Muga, who is now doing her PhD. And we went with Mama Mina, and she said, I think this plant is sick. And we said, cool, let's take a leaf. Let's analyze it. Let's see if it is. And so we did this all over the place. Here's some of the devices. The machine in the middle is the DNA extraction machine. My friend Jo Stanton, who is in New Zealand, she's an amazing scientist who developed this machine, and it runs off of a motorcycle battery. And it's called a PDQEX, which stands for pretty damn quick extraction. So she, through her science in there, and we controlled it by a mobile phone. So one of the biggest challenges is getting decent quality DNA in the field, right? That's a huge barrier. And so we use this really cool device that was hers. We had a big group of people who were doing library preparations. I mean, lab work looks cool in the lab, but farmers were like, what is this mumbo jumbo? That we were wearing gloves in the field, which always made me laugh. Here's some of the technology. It was all portable genomics things. You have the DNA sequencing machine in the background, and then we have our little mini supercomputer there, which is called the MinIT, which has some NVIDIA really cool GPU graphics to do some of the analysis, and our little sequencer in the middle. And we ran this all off of a laptop power bank. So we did this as a trial, and we didn't really think about it. So the thing I love about this picture is the women were drumming for us as we were doing our analysis, and they gave us their drum afterwards to sit the devices on. So that was such a cool thing for us. And one of the most important things I want to say about this is we went from 6 months for somebody to diagnose the plant, right? Typically, the scientist would grab the leaves, send it to a lab in Switzerland or Korea or the UK. And then 6 months later, you may go back and tell the farmer that there was a problem. And the most beautiful part of this is it took us 3 hours to tell. Our outreach was a little they're holding my laptop, for crying out loud. We didn't really think about, this is going to work and what are we going to do when we tell them? But they were, the thing is, they're happy to get the results, right? Farmers just want to know, early detection is key. If the plant is sick, I need to get it out of there and get something in new for my family, right? So these farmers were really happy that we had told them their plants were sick, because now they could do an intervention and save and not have a hunger season for their family. So that was that. The next slide is a video, and I want to just give you, it's just the background. It's just the farmers talking about what this meant to them.
See the results right now. Yes. See the results right now. Yes.
Asha has too much cassava. And that's a really Asha has too much cassava. And that's a really great problem to have.
Na kuzalisha mazao mengine unaweza ukategemea kuuza muhogo, ukaenda kuzalisha mazao mengine unaweza ukategemea kuuza muhogo, ukaenda kununua mchicha, ukanunua mbegu ya maboga, ukanunua mbolea, ukaanzisha bustani.
Good quality DNA. For me, the biggest part of that video is the fact that she had enough to sell and she's actually building a house for her family. And that to me was like, that's real science. That's science actually, not like us using buzzwords or whatever, you know what I mean? But actually transforming a community's existence. It meant everything to me. It makes me sometimes a little emotional. I'm not going to cry because you're recording this, but it makes me feel some kind of way. Anyway, as scientists do, you know, just giving a TED talk isn't enough. Haters going to hate. So we wrote a paper. There you are. There's the paper. It's peer reviewed. You can go get your little things in there that you need, but we did our thing. And we made sure that, you know, we dropped the paper on the same day I gave the TED talk, because there's all this thing like, all you do is talk and you don't, no, there's your paper, people, go cite it, do what you got to do. Anyway, there's that. And I just want to say that, you know, I've sort of transitioned to not leading the project, because I think that one of the most important things that I could do was know when to step out, right? Because I'm not African. That is not my thing. We got the project going. Now East African scientists are leading it, doing amazing things. And I am more of an advisor now. I feel like that was a really important thing. I love it so much, but sometimes you have to love it enough to let it go, to let it grow. And things that support for Africa, the money actually has to go to African scientists, not me, right? I mean, you just have to make those hard decisions. So I now work for BioTeam, which is a scientific consulting company. And we also have a project where we're trying to build computational infrastructure, because through that whole process, I realized that analyzing big amounts of data with no internet and no power is a problem. So I've sort of now come back to the US. I've just moved back to the US from Uganda about, I think, almost a year ago. And I'm really enjoying now this collaboration and trying to work on other forms of technology to partner with teams in the region. So we're working with this ACE, which is African Center of Excellence in Bioinformatics in Kampala, Uganda, to try to find solutions for analyzing data in really low resource settings. So we're working on that as well. And I'm really enjoying that. And I'm really loving seeing the Cassava Virus Action Project grow within East Africa as me as an advisor. So that's what I'm up to now. And of course, as I said, it's been hundreds, maybe thousands of people who have helped us do what we did. And I'm really grateful. And here's a bunch of people who have said, who I need to say thank you to. And most importantly, the farmers who trusted us to burn their fields and replant things that were tolerant of the diseases. So anyway, that's me.
How inspirational. Both of you. What amazing stories. Laura, let's start with you for 1 second. So, I mean, you were told, as you said, by people in the UK that this was not going to work. What were the biggest barriers for you to getting to what you wanted to have happen? And how did you get over them?
Oh my gosh, it's such an interesting question. I think the biggest barrier. Okay, well, first of all, I'll just get straight to it. Number one, the technology is fairly new. And how in the world is that ever going to work on the African continent when we can't even get it to work in our lab in the UK? How will you ever do that? It's so new. It's so hard, whatever. So that was one. Secondly, if you think about the amount of pushback that I got for it, you got to realize that there is millions of dollars on the line here. Let's be real. Scientists write grants and the sequencing budget is huge. We've just introduced a technology that removed a big portion of the grant, right? Africans don't have to partner with you anymore, nor are they going to. It's about empowerment, right? So I just want you to envision the pushback I got from colleagues about democratizing sequencing, how that went. I'd love, we can talk about that at some point, but it was massive, right? And it's always based on money. Always. It's always based on money, because when you empower people, it doesn't go well for the person that thinks that they're being sidelined. But in fact, you're not, because everything is going to get better. When everybody has access to the thing, everything would get better. And in fact, one of the things I realized when, the whole SARS-CoV-2 thing happened was, man, wouldn't it have been cool if we already had empowered people to be able to collect the genomic data, and not just from South Africa? We have no idea what's circulating in Tanzania. Not a single, and it's not because of capacity, it's because of mistrust. We have no idea what's circulating there. We don't. Check all the repositories. They're not sharing their data because we didn't do it right. So anyway, that's a whole nother thing. But the biggest challenges were nobody's done it before. And I'm like, they're sequencing in space, for crying out loud. Why can we not do this on phone? And you know what? Also, honestly, I don't care if I fail, whatever. We tried. There's no other option, right? They don't have another option to get the genomic data to figure out what's killing the crops. So like, okay, we fail, whatever. None of us were afraid to fail, truthfully, but the pushback was pretty big. It was massive. And we just said, oh, well, we're doing it. And we did it. And the funders in the game, I'm not going to mention them, but there's huge funders, nonprofits. They've doubled down on regional sequencing hubs. So we got a lot of pushback from the big funders as well to not do this, but you see, we did it anyway.
Laura, don't you love it when scientists say it's never been done before? Well, it wouldn't be science if it had been done before. I always think that's code for, that means a man hasn't done it before.
Exactly. Oh my goodness. Yes. No. And can I tell you truthfully, the person who bet me 2 bottles of champagne we couldn't do it was a woman, a white woman. I'm like, well, you know what, lady? Where's my champagne? If you're watching this, where's my champagne? People, it's fascinating. I get it. It's scary to do something, but what is the other choice? Like, there's no other choice. We need these farmers. These farmers should have access to the sequencing technology and results quickly, just like any other place in the world, But yeah, I do love it. I love when people tell me I can't do stuff. It's my fave.
And Jo, you have a very different experience, but I'm wondering if you were saying earlier how in a new antibiotic discovery, the big companies got out of it, and you had said it was driven by, it's not going to be as profitable. How much in your career would you say has had the same influence of kind of the money not, driving those decisions? Or do you think there's other things at play in the experiences that you've had?
I think there are a lot of things at play. One of them is being a woman and having non-traditional ideas and unconventional ideas. When I first introduced the concept of metagenomics, which is the idea of collecting DNA from soil or any environment directly, extracting it without first growing the bacteria, people said that's ridiculous. That can never work. And it hasn't been done before was a classic. And I always saw that as code for if a man, hasn't done it, then it can't be done. And I don't know if I would have been successful in that field if it hadn't been for a couple of men who came to the same ideas kind of simultaneously with my lab and made the area, helped make it famous. Somebody very, very famous like Craig Venter, who, sequenced the human genome and then went on to environmental sequencing. And, he didn't want to use the word metagenomics because I had come up with it, but eventually he did. But that of course helped everybody in the field get the concept accepted. So I think being a woman has been problematic. The antibiotic area, particularly the industrial side, is extremely male. And there I also had trouble breaking in, it's sort of this attitude of, well, if, we can't come up with new antibiotics, what makes you think you can do this? And I can give them a list of 8 reasons that we're doing things differently than the way they did them. And I still get the question, why do you think you can do this? And I say, well, because I have science on my side. So I think that the money is part of it, and certainly the investment in antibiotics faded away largely because there's so many drugs today that are so much more lucrative. But I think it's been especially hard to convince people to remain in the antibiotic field or to support the work that we're doing. Because one, it's unconventional, it's crowdsourcing, it's democratization of science, which is, anathema to the industrial kind of science that's so common in our field. And it also is led by a woman.
What do you think, if anything, has changed over the last several decades for women in STEM? Has there been change, do you think?
I think there have been a lot of changes. What is sometimes remarkable on the bad days is how much has stayed the same and how we still hear the same negative messages, and women are still struggling with exactly the same issues of the imposter syndrome and combining families with being scientists. But just having a lot more women in the field has made an enormous difference, because there's a variety of role models, and there's a lot of research showing that people have a hard time doing what they have never seen someone do. And so, women who only saw women who didn't have families, for example, be scientists, didn't have a role model if they thought, well, I want to have a family. So they would choose to leave science to have a family. And it took a long time before we had enough role models to be able to say, no, you can do both. And it's been done by both men and women really successfully. So I think the role models is a really big part of it. I think where we're absolutely lacking role models is with underrepresented groups other than women. And the intersection of being Black or Latinx and a woman, I think, continues to be an enormous challenge in science. So I think a lot's changed, and just having more women, having more research to cite about women. I'll just quickly tell one story that for quite a while when I was working on an NSF project on women in science, I gave talks about unconscious and implicit bias. And every single talk I gave, somebody, male always, would say, but we're scientists, we're objective, so we don't have those biases. And I would use all the arguments that I could pull out of my arsenal from, well, we do blind experiments because we have bias, right? There's some reason for that. So maybe we do have bias. And these are unconscious or implicit biases that we're not aware of. So how could we possibly say we're not subject to them? And we come out of a society where everyone has these biases. None of that worked. And so finally, when I was at Yale, I had a postdoc who had a background in this field of psychology. And I gave one of these talks to a group of department chairs, and they all said the usual, but we don't do that. We're scientists. We're trained to be objective. And I said, and I gave them all the data, and they said, yeah, but those studies aren't with scientists. And I got back to my lab and I was talking to my postdoc and I said, I think there's only one solution here. And she had come to exactly the same conclusion, that we just have to do the experiment on scientists because they think they're a different species. So we did, and we sent out a resume of a student, and half of them had randomly assigned the name John and half were randomly assigned the name Jennifer. And we got reactions, and the short version of the story is that people were, men and women, a lot less likely to hire Jennifer. They were less likely to mentor Jennifer. They'd pay her less if they did hire her, and they thought she was less competent. And it was the exact same resume. So that, I think, changed the conversation a little bit, that no longer could scientists say, we don't do that. Well, these were real scientists in real science departments and real universities who did exactly that. So again, I think the research has shored up our position to convince people that these are problems, these are issues, and we can fix them with science.
We heard about that experiment in our hallways, and it definitely made it to so many different areas in the scientific community. So yeah, thank you for doing that. It was just an unbelievable experiment to do and results. So now, and Joe, you've had the ear of the president of the United States. Laura, we mentioned you have a TED Talk that's widely viewed. So in addition to those things, how else can people be communicating what we're talking about here today and making changes, going back, our audience, going to their own departments? And like, what advice do you have for people for communicating these things that we all feel so passionately about?
For me, I don't know. I just feel like we have to step away from like white women as the gateway drug to diversity, right? Like we just have to get that out of our heads, that we are somehow the diversity. Like we have to step down when opportunities are there. We have to step down and we have to do the work of nominating people, right? Like we can't, I have myself a little Twitter rant this morning, which I'd never do, but I wanted to today because I was, taking a shower as you do. And you're like, I'm not surprised by the vaccine inequity at all. Mm-hmm. Science is racist, people. How many African scientists did you really have on your keynotes, on your papers, on your grants, on your editorial board, on your author list? What did you think was going to happen, world? The whole system that designed the vaccines and did the things wasn't inclusive. So here we are. So anyway, that was my revelation this morning. Cause everybody's like, cause we're all never going to get out of this till we're all out of this, right? We have no idea what variants are circulating in places that don't have sequencing. That is a fact. Okay. So that's the bigger picture. What advice I would give to people: do the grind, do the uncomfortable things. If you have any position of power, do the hard work and give the keynotes to people that deserve to have their voices heard. You're not going to be popular, but too damn bad. Keynotes, publishing, editors, opportunities to speak, grad students. I would love to redo Joe's experiment and put like an African name at the top versus like, I'm focusing on Africa because that's what I'm most familiar with, but imagine how that looks. And so for me, the TED Talk is about the science, but it's also, everything is visual about that, right? Every single thing that we did as scientists, that nanopore device was only held by white hands. We had to take those photos to make it, it's this role model. Like if you see only one type of hand holding a thing, is it for me? And I think that it's just every single scientist and every single person sort of floating around that space can really do the grind every day. Little things add up to a big thing. If an email comes in and you're invited, okay, who can I, and saying the thing when you're on the organizing committee or the advisory board or the whatever, the uncomfortable thing of, oh, do you think we have this right? Oh, I don't know. Maybe we should rethink. So for me, it's those daily things. And look at, everybody loves scientific societies so much, but do a good hard look at them. And your DEIA thing on the side is not the thing. It needs to be embedded in everything you're doing. You don't have that as a sideshow and then do your keynotes and have them line. No, that needs to all filter in. So that's my thing. I stay on the grind trying to do those things, and let's see how we go.
Joe, do you have anything to add to that?
That I think I couldn't say it as well as Laura did, that, you do have to stay on the grind, and it's exhausting. It never stops. And that, I think, is demoralizing. But when you're right and you start seeing small changes, that's what keeps it going. We started in my institute a new group called AJEDI, which stands for Anti-Racism, Justice, Equity, Diversity, and Inclusion. And we wanted to bring anti-racism into the typical DEI to sort of elevate it and get it up front and get people used to the word, of anti-racism, that this is a thing you can do. And what has struck me is how many practices there are that people either aren't aware of that are anti-racist, or that simply make a more equitable environment, or they just don't think to use. And if you highlight it, you make it visible, then other people will start doing it. Because the fact is, most people do care about equity and fairness. It's just they don't know what to do about it. And I think the other place that we need to focus is in the classroom, because in STEM, we lose most of our diverse students before they graduate. They start out with talent and interest, and we get rid of them very effectively. And we have to change the practices that are embedded in our institutions and in our classrooms that are absolutely outright racist and discriminatory.
Absolutely.
Joe, just drop the mic. You just dropped the mic, Joe. Love it.
So, Joe, as a microbiologist, I want to ask you, what has been so fascinating about soil microbiology for you for so many years? And I also want to ask you, what prompted you to write your book now?
Well, microbiology and soil, I think it's the diversity that I love. And there's always been this parallel of, I like studying human communities and diverse communities in humans, but I also like the same in bacteria. And when I first started out, I knew these microbial communities were so diverse. The soil is the most diverse environment on Earth. And as I've lived through my career, it's only become more diverse because we have better tools to see it. And that diversity and that extreme complexity is just a source of endless fascination. But what has really been exciting is that when I first started, I remember as a graduate student saying to my advisor, what would be so great is if we could see all the genes and all the proteins and all the RNA. And I wasn't talking metabolites then, but I probably should have. If we get a picture of all of those, maybe we could build models. I probably wasn't even using that word then, but my idea was you could build predictions, and today it would be through modeling, about what communities will do if you had all that information. And when I came back from the White House, we finally had the tools to do it. And I waited many decades to be able to do that. But it's now the most satisfying part of my career, because we absolutely can sequence all the genomes of all the organisms in the community. We can figure out all the transcripts and what turns them on and what turns them off. We can detect the proteins. We can detect the metabolites. So for me, this is like the realization of the science I always wanted to do. And the technology has given me that gift of being able to see the pictures of communities that I want to see. What drove me to the book was I discovered when I was in the Obama administration that I'd been trained as an undergrad in soil science, and I had worked in soil microbiology my whole career, but we all get so kind of focused on our own little deal. And I was studying my microbial communities and antibiotic production and the effect of microbes on plants. And I just sort of had my blinders on. And what I discovered when I started looking, when I was working for President Obama, on what do we need to do to advance soil science, was that the soil erosion crisis, which when I was in college, everybody thought we had conquered. We're not eroding soil anymore because we have all the tools. In the decade I came out of college, no-till farming had just started, where you don't work the soil over, which is one of the biggest sources of erosive activity. And so everyone thought, well, the farmers are just going to switch to this and we won't be eroding soil and all will be fine. And what I had missed was that the legislation that protected soil just kept getting eroded and eroded to the point that USDA, which has the mandate of stewardship of soil, just couldn't enforce and hold farmers accountable for their plans to protect soil. And so I realized, and I got tutored by many scientists across the country, that we were in a major crisis of soil erosion, that we have lost, for example, in the Corn Belt of this country, which is the most productive soils we have, we've lost about half the soil we had at the beginning, and about a third of the soils have no topsoil left. They're farming on subsoil, which is not very productive. And you go across the world and you look at Nigeria and India and just every country, Ukraine, every country is losing its soil. And I realized that we were facing a crisis that would probably have the potential to kill us faster than climate change, because we won't be able to raise crops in another few decades. And without crops, if we don't have food, then climate change won't be what'll get us. And so the combination of climate and soil turns out to be a really fascinating and I think frightening area. But the good news is that we can fix the soil problem. And that's, I think, very satisfying. It's very hard to fix the climate problem. We know exactly what we need to do to fix the soil problem. We just have to have the will to do it.
Okay. And in our last couple of minutes, I have a question for both of you. And so, Joe, I know you used to speak to the President of the United States. I don't know if you still do speak to the current President of the United States, but if you could, and Laura, same question, what advice would you give this administration for the moment that we're in right now, which is, unlike any other moment really in our history? As far as not just about the pandemic, about science and diversity, about the pandemic. Well, if you had 30 seconds with the president, what message would you
I've
share?
written a couple of policy pieces for Biden that I don't know if they will ever reach President Biden, but they have reached other people, and that's exciting in government. And one is soil, that we should be supporting practices and encouraging farmers with incentives to practice good soil stewardship. And some of that has actually started to enter the current budget and the Build Back Better bill. And on the other hand, we need to be empowering women and minorities in science. And I focus largely at the college level. I think we need research courses and we need active learning and the ways that we know will support learning by diverse people. Those are my two recommendations.
Laura?
Oh my gosh, I would love to do this. Oh, it'd be my dream. Number one, they need to travel to Africa, East Africa, get on the ground, do some things. Number two, they need to get control over the family not-for-profits that are funding things to do things. Individuals cannot drive policy, period. They're going to dangle their money. You got to say no. So my thing is hire more Africans, go to Africa, and tear down all of academia and start over more inclusively. How's that? I can't stand it. Toxic and nasty. And I know we're sponsored by publishing, but let's have a whole sesh on that. But anyway, I would say that we're just in this current climate of COVID, we are not going to get out of it until we start making things more equitable with access to the scientific tools that are there. We are never going to get out of the COVID thing until we know what's circulating around the world, period. And we're never going to get out of it until you greedy get your vaccines over there. The end for me.
Okay. Well, and that will end our discussion today, and we'll conclude on that note. So I want to thank both of you for joining us today. It was a terrific session, a terrific conversation, and I know I learned a lot. So, and to our audience, please stay tuned for our next session in the program called Precision Medicine, Future Medicine, where I'll be talking to Kenna Mills Shaw from MD Anderson Cancer Center.